Okay, I think we're good to go. Yeah, so today I was going to continue from where we left off yesterday, right? So we'll try go over some of those concepts again, and I think it'll be a lot easier today after Kavita's lecture, right? Because connecting the theory to something very applied might make all of this a lot more seamless. But before that, Kavita mentioned Francis Galton, and he's a pretty infamous figure in human genetics, right? Because he was Darwin's cousin, but he was also a raving eugenicist, right? He was also one of his most famous papers is in Nature, where he went around like rating women on how beautiful they were in different parts of England, and sort of pricking his finger to sort of rate them from 1 to 10. And the thesis of the question of the paper was which parts of England have the prettiest women, and this was published in Nature, so Nature was a very different journal back then. Okay, but his most perhaps useful invention for this audience is what's called the Gumpton reviver. Anyone heard of this? Okay, so this is how he described the Gumpton reviver. It's a funnel with water that drips onto your head as you're reading a book to prevent you from falling asleep, right? So that was a funnel dripping water which runs off and is drained appropriately so students do not fall asleep while reading heavy books or while listening to lectures about GWAS, right? Okay. Yeah, before I move on to GWAS, I wanted to sort of touch upon two concepts that Deepa and Kavita mentioned before. One was, Deepa yesterday, spoke about synonymous mutations, missense mutations, loss of function mutations, right? DNDS. In humans, we have these million genomes, right? We have hundreds of thousands of genomes, so we can look at the number of synonymous mutations, number of non-synonymous mutations, number of stop, sort of nonsense mutations per gene, right? And we can observe the patterns to try and predict which genes are likely to be most important for function. Okay, so this is from a database called GNOME-wide, which is the largest available public database of human genomes. I have sort of pulled out data for two genes. One is CMIP and one is an olfactory receptor, OR284. What I want you to look at is this, I'll use this slide, this projector. Okay, so here is for the number of synonymous variants you see. You have the observed number of SNVs, of sites that vary, right, in this gene. That's 259. You also know what's expected based on the length of the gene, based on the sequence context, right? We can build an expected model of what's expected, what's observed, and O by E gives you a sense for, okay, do we see as many mutations or as many variants in this gene as we would expect by chance under a neutral model of evolution, okay? So that's the O by E for synonymous. That's the O by E for loss of function variants. And similarly for the olfactory receptor, this is the O by E for synonymous variants. This is the O by E for the loss of function variants, okay? How do you interpret this? What do you think is going on? Which of these two genes do you think is likely to be important in severe disease? Do those numbers make sense? Did those, yeah? Okay. How many of you think the olfactory receptor, the second gene, is more likely to be really important in disease, in severe Mendelian kind of disease? Why? Because it has more loss of function. 4 compared to 2, is that what you mean?
No.
Then?
O by E is 0.75.
O by E is 0.75 and O by here E is, okay, so this means that you observe 75% of the mutations sites that you might expect to be mutated are mutated, whereas here 5% of the sites that you might expect to be mutated are variable. Do you still think that the olfactory receptor is? Does anyone have guesses on which gene is more likely to be important for disease, or really important for organismal function in humans? Why? It has more missense. Tell me what you mean by that. Tell me more about what you mean by that. Also, a hint, I think what would be helped the most is to look at synonymous and this one, the last one, the loss of function one. That's usually like very informative right away, but okay. What were you thinking about missense? Okay. Yeah, missense is informative. I'll come back to you. Anyone else would like to take a guess before I make the big reveal? Okay. Okay. So this gene, the CMIP gene, is the more likely to be it's likely to cause a disease if knocked out or if it doesn't work, right? Because, okay, so first let's look at the number of synonymous variants, right? Synonymous variants, despite there being some kind of, no one contests that they have some kind of function, but they're unlikely to have a severe function, right? Which is why you observe about as many synonymous variants as you expect. The observed divided by expected is like slightly higher than 1, but close to 1 for both sets of genes, right? Here it's 1.22, here it's 0.94, but the confidence intervals are overlapping 1, okay? But let's look at the nonsense mutations, right? Here, you observe a lot fewer nonsense mutations than you expect by chance, which means that a lot of these nonsense mutations in this gene have been selected out, right? So you don't see them in the human population because the people who have them would not exist, or there is just a high degree of selection acting against carriers of that mutation. Whereas for this gene, the observed by expected, it's still lower than 1. So there is, well, but the confidence intervals are very wide. It could be 1, right? If you take the error bars into account, the number of observed nonsense mutations is actually consistent with the neutral model of mutations. And therefore, this gene is unlikely to be functionally super consequential for disease, whereas that gene is way more likely to be involved in some kind of a severe disease. Does that make sense? Does that not make sense? Okay. So I wanted to highlight sort of the kinds of things you can do within populations, right? Not just cross species, but within a single species by sort of looking at a large amount of data from within individuals of the same species. Okay. The second thing that Kavita had pointed out today, no, Deepa had still pointed out, I think today or yesterday, was the mutational spectrum, right? It's actually, this is just an anecdote, but this paper in 2017 looked at these genomes from multiple different populations, and looked at whether certain populations have different mutational spectra, okay? So here, we're looking at mutational classes. So we're looking at a mutation, let's say a C to T, but also looking at what is the base before and what is the base after, okay? So we're looking at trimers of nucleotides, right? So for example, this cell would be an A to T mutation, but a C before in the 5' end of the A, and a C after, okay? So a CAC mutation to a CTC mutation, okay? And the colors represent, is that mutation type different between European populations and African populations? So the redder it is, the more likely it is to be significantly different, right? And I don't know, my hypothesis would have been that you really wouldn't see my hypothesis would have been that we would have seen all blues. Like, why would the mutation spectrum differ in a species that's relatively young? Like, why? I can't imagine selection acting on mutation rate. Like, what are the reasons for that, right? But what you see here is a lot of reds. So a lot of kinds of mutations seem enriched in certain human populations compared to others. We don't know why yet. This hasn't been, yeah. Why?
Older than the...
Yeah, but why would the mutation rate why would the mutation bias, I guess, as Deepa said, change, right? You would expect a difference in the number of mutations, maybe the number of variants they carry, and that we do see. But why do we see more, for example, Europe shows the highest, sort of the most significant difference is I think TC TCC to TTC mutation? Like, why would that be different in Europe versus Africa?
Very different environments.
Yeah, so that's I think being looked at. We don't have a causal evidence, right? Intriguingly, that signature, that TCT to TTC, TCC to TTC signature is also the signature that occurs because of UV damage, DNA damage from UV rays, and we know that Europeans sort of have had a whole bunch of selection go on with directly because of less melanin in the skin and therefore more exposure to UV. So is there a connection there? Why would that be causal? We don't know the story yet, but this was sort of the first sign that there might be a story there.
South Asia versus East Asia.
Yeah.
So it's more whitish as compared to Europe versus Africa. Is that right?
By more whitish, you mean it's not, we have fewer differences. Yeah, we do seem to see fewer different. The strongest, I think, the signatures are here, right? That red block there, yeah. So this is fairly new. It's not explored very much, but this was a result that really blew my mind because completely unexpected. I would not have expected to see such stark population-specific differences. Yeah. Okay. So Deepa had spoken about like this. What are the different kinds of mutations? Can this be raised up? Is there someone there? Okay. Well, okay. You can have an A to G mutation, right? But they didn't just look at A to G. They had the power to look at A to U. Even within this kind of mutation class, you can have you can split this up into like 16 different, right? Whatever. Essentially, they split this up into each of those different kinds of mutations, taking into account the surrounding bases, right? Because mutation rate can also depend upon the genomic context, right? Does that make sense? Okay. Okay. So yeah, that was another thing that I wanted to sort of just highlight as a connection to what Deepa spoke about. Yeah.
I want to focus on South Asia versus East Asia.
Huh.
If environmental factors, these are the factors.
Yeah.
Then these countries are close, right?
They are close. Yeah.
So how can the environment?
They are close now, but I mean, East Asia, they have they're close now, right? But historically, environments could be different, cultures could be different, exposures could be different. This is really completely underexplored. We have no idea why. What are the underlying causes of something like this, right? It could be exposure not just to the environment, but it could be, for example, melanin, right? East Asians have different amounts of melanin on their skins compared to South Asians. So something that, again, could point to that TTC to TCC signature. Yeah?
But the T one is more darker, right? That T panel, if you compare with the Europe, yeah.
Yeah.
So that is not there in East Asia and Africa.
Yeah.
That would be a reason of this thymine dimers and all because they are exposed to a lot of UV radiations and that.
You should hypothesize, and maybe, I don't know, all of you have come up with projects already, but this might be another project for you to hypothesize about. Yeah.
How is the difference between the mutation spectrum between the populations calculated? Like, what is the scale?
This is the p-value, so this is a significant scale, right? They essentially think about it, right? In each population, you can calculate the number of times each kind of mutation occurs, and you're trying to say, okay, is the number in Africans correlated with the numbers in Europeans, right?
So is that a correlation of sorts?
No, this is a p-value, I think, this is the minus log 10 of the p-value, right? Which is why the more red it is, the higher the values are, yeah? So think about it as a significance measure of sorts. Okay. Okay, so, but yeah, feel free to read that paper. It's a very fun paper to read and look this up. Okay, but let's go back to GWAS and where we were yesterday. So Just want to highlight that GWAS is used to study common variation that we capture using tag SNPs. This is not done using sequencing. This is, the experimental design here is intended to be an alternative to sequencing, a cheaper alternative to sequencing, okay? And we do this using microarrays. We can run a GWAS either on a binary trait, so diabetes or not, pick a disease, like a disease status versus not deceased, or on quantitative or continuous traits, so like Kavita said, height, cholesterol, pick your favorite trait, yeah? Okay. Before we go into the steps which we started yesterday, let's spend a little bit of time connecting GWAS to population genetics, okay? So I want you to think about these two dimensions of variation, okay? So one is allele frequency, right? Minor allele frequency, very, very rare. So these are mutations, like rare mutations here, to very common variants, right? Variants that you see in about half the population. And on the y-axis is effect size. So what is the effect of a variant on the disease? Over here, these are very small effects. So if you have the risk variant, your height might just increase by 0.1 millimeter. Whereas if you have a variant that has a very large effect on height, your height might increase by, I don't know, by 10 centimeters, 20 centimeters, whatever, right? So effect size, small to large. Okay, so you're a clinician or you're a human geneticist, okay? And you want to find, you care about your favorite disease, you want to find genetic variants associated with your disease. Where do you want those variants to lie? What do you think would be the most helpful in terms of predicting whether a person has a disease or not? Or, yeah? Go ahead. Yeah. Which quadrant, right? There's four, like divide this into 4 quadrants. Rare, common, small effect, large effect. Which quadrant do you think you would want to see your variants in, huh? Large and rare. Okay, so let's say you find variants in this quadrant, right? Rare and large. If they're rare, they're not going to be helpful for a lot of people because they're rare by definition. So these variants will help you find disease, in maybe 1 out of 1,000 people, 1 out of 10,000 people. But for most people, they won't be that helpful, right? Okay, so you might want to find variants that are common, and if they are found, they automatically explain disease, right? So they have large effects on disease. Do we find them? Can we find these in GWAS? I mean, Kavita's already told you the answer, but so this is a test. But yeah, so can you find those? That's what we want to find. The whole field of GWAS started hoping, okay, let's try and find variants in this quadrant. We will solve all of genetics. One of us will win the Nobel Prize, whatever, right? But do we find these? Do we find variants there? Do we find these common variants that have very strong effects on disease risk? No? Why not? Yeah, so they do have small effect size, but why? Why do they have to have small effect? Why can't we? Why can't they be here?
If you have a large effect size, you are likely to be selected against.
Exactly, right? So selection does not allow variants to be found in this quadrant. If you have variants with a large effect size, selection will push them to be rare. So selection will push them towards this quadrant to become rarer, right? So that's why it's next to impossible to find that class of variants, okay? Okay, so what GWAS finds then is this quadrant of variants. Common variants, because that's what we test for, that's what we design those chips for, with small effects on disease, okay? Other kinds of studies end up finding sort of these rare class of variants, either with large effects or small effects, right? Rare variants with large effects are still helpful in family studies, right? If a family comes to you with a very rare severe disease, you can find these rare variants and help that particular family, and then other studies may help you find other mutations in that same gene, so on and so forth. But it's a different class of sort of experiments, right? You use sequencing for this. You don't use GWAS for that, because GWAS chips would not capture these rare variants. GWAS chips are designed to capture common variants, okay? Okay. Everyone with me so far? Okay, great. Okay, so we went over this yesterday, but a quick recap again. So let's say you care about diabetes, your first step is to collect your sample, cases, controls, collect DNA, genotype each of them at 1 million tag SNPs, and what you get is something like this, where for each SNP, where M is 1 million, you get a genotype matrix, right? And here we label our genotypes as 0, 1, and 2, right? Yeah.
So not diseases, but GWAS can find large effect loci also because I was looking at this Galapagos finches.
Huh.
So for that, I think now they also find large effect loci.
We can, if
That's using GWAS. That's
if-
possible to do, right?
if selection is not acting against them, then you could find those variants here, right? So the reason we don't find these is because of selection. But yeah, because of negative selection rather. So I think one of the largest effect GWAS variants in humans has been for Alzheimer's, which is a late onset disorder and therefore less selection acting against it, right? The ApoE gene. So how much you find there depends on the degree of negative selection. Yeah. Okay. Okay. So this is what our data look like. Our data from our GWAS look like. I know one point that I didn't make very clear yesterday was sort of how analysis differs for whether your phenotype is binary or continuous, right? So let's say it's a binary trait. Your sample collection, genotyping, everything is the same. What differs is how you run your statistical test. So the kind of test you run is called a chi-square test for a binary trait. The kind of test you run for a continuous trait, which we saw, which Kavita also mentioned, was a linear regression, okay? Each of these tests will give you a p-value and an effect size. So it's not like one of them gives you that p-value and that effect size. Both of these kinds of tests will give you a p-value and effect size, and then you pick the ones that are significant based on your p-value threshold, okay? Okay, I know I had gotten a question about chi-square tests yesterday, so this is what it looks like, right? You have two alleles and cases and controls. So a chi-square test essentially makes a 2x2 table and asks, is there a sort of is this table not proportional? Is it tilted? What do you think? Is this variant associated with disease or not? This is a toy example, right? So let's say, yeah, I have created these numbers for a particular variant with two alleles, capital A, small a. Is this variant associated with disease? How many of you say yes? Those of you who don't have an opinion, I'm going to ask you for why, right? You know that. So, okay, how many of you say no? Okay, nobody raised their hands at all. Okay, what do you think? Yeah. Why do you think, okay, so you think it is associated with disease, why? Can you speak a little louder? Yeah.
The control case has
Yeah.
way greater a number as compared to the case.
This just means, okay, the total number of controls is more than the total number of cases. That just means we have sampled more controls than cases, right? So comparing these two columns doesn't tell you much. What helps is comparing. So for capital A, most carriers of capital A are controls, maybe about maybe twofold, right? Whereas for small a, it's 24-fold. You're 24-fold more likely to be a control than a case for small a. For capital A, you're twice more likely to be a control than a case. So it's 24-fold versus twofold. That's the comparison you're making. And that's how I would say, okay, capital A is way more likely to be a risk allele for the disease compared to small a. Make sense? Yeah?
Could you repeat that last sentence?
Yeah. So what we're conceptually, there's a formula for this, but what we're intuitively, conceptually trying to get at is for capital A, for carriers of capital A, you're twice more likely to be a control than a case, right? Whereas for small a, you are 24-fold more likely to be a control than a case. right? So that number is huge. These two numbers are hugely different, right? And therefore, that gives me I would assume that capital A is a risk allele associated with the disease, okay? The sort of statistical way to compute that is a chi-square test. And the equivalent of the effect size from a chi-square test is called the odds ratio, where you're essentially multiplying these two and then dividing it by the product of the other two cells of the matrix. I'm happy to talk about this after class or in the tutorial, but intuitively it captures what I was talking about just now, okay? A chi-square test doesn't allow you to use covariates, right? It doesn't allow you to correct for age, sex, other things that you might want to include in your model. So in practice, what we use more often is something called logistic regression, which is like a linear regression, but for binary traits, which allows us to sort of say, okay, I want to take the effects of sex into account, of age into account, of any other phenotype into account as a covariate, okay? Everyone with me? Okay, so that was that. What do the results look like? This is what the results, a cartoon of what the results of a GWAS look like. Okay, so if you remember, we run 1 million tests, one for each SNP. If you want to visualize the results, we plot each of those results one by one. So here we are, on the x-axis is all SNPs sorted by position by chromosome, right? So 22 autosomes here, we're ignoring sex chromosomes. Y-axis is the minus log 10 of the p-value, so the higher up, the more significant, okay? The red line is that threshold for multiple testing. Anything above this line is considered significant, okay? This is called a Manhattan plot because when it works well, these peaks are supposed to sort of represent the skyscrapers of Manhattan, but if you're unlucky, you don't get any peaks and then it just looks like a Bangalore plot, I don't know. Pick a smaller city, okay. Okay, so what this result represents is a GWAS for which you have one strong signal, right? This set of SNPs seems to be above your significance threshold. The reason you see a peak like this is because all of these are they're not tightly linked, but they're sort of linked, they are in linkage disequilibrium to some degree, right? Because this set of SNPs is close by on the chromosome, and that linkage makes their p-values correlated, right? So that's what that peak represents, okay? That's what leads to the peakiness of these Manhattan plots, okay? So what I want you to take away from this is that this represents one peak. This is not 5 different signals, but one signal, even if the number of SNPs that are significant is 5, okay? All right, so let's look at an actual GWAS. So that was a toy example. This is a GWAS from about 4 years ago. So now the height GWAS is way different, but let's look at this one. The first thing to notice is that the sample size. So this was a GWAS that worked with 360,000 individuals, measured height of all of these people. The reason we look at height is because it's the easiest trait to measure. Not because, I mean, it happens to be biologically interesting, but it's because you can measure height for anyone very easily. Okay, so you have 360,000 individuals, which is a sample size. First thing to notice is that, look at the y-axis. This is close to e raised to the power minus 292, right? We're reaching, like the statistical significance levels we're reaching are insane. We have the power to pick up really tiny effects, really tiny associations of your variant with your phenotype, okay? Also, look at the number of signals you see, right? Polygenicity. This is, like, you can't get more polygenic than this, right? I think if I remember correctly, there's more than, 12,000 independent signals associated with human height. So there's 12,000 positions in your genome that have some influence on human height. And so this kind of plot sort of generates a lot of work for experimental biologists, right? To go and look at and figure out what those 12,000 regions are doing. Yeah? Okay. This was perhaps, more important of the last few years, where they ran a GWAS for severe COVID, sort of looking at what are the traits in the genome making you more likely to have severe COVID, and this was much smaller number of cases, right? So 13,000 cases, and they found these signals. I think the interesting anecdote from this was that this particular variant, which has the highest effect size, the highest significance here, but also the highest effect size on severity of COVID, had a high allele frequency in South Asians. So that was something that made Indian public health experts sort of sit up a little bit. Okay. Okay, so why are we doing all this? What have they told us? Has this all been, like one question I get asked a lot is this just a gigantic waste of money, right? Like what are we going to do with all of this? So what have GWAS told us? Firstly, traits are highly polygenic. There's hundreds and thousands of variants associated with any trait that's been studied really, right? So with cholesterol, there's now more than 1,000 traits associated with cholesterol now. And the cumulative risk, genetic risk of an individual is going to be the additive risk, the additive effect of all of those associated variants, right? So if there are these GWAS that have been conducted, based on those GWAS, I want to predict my risk of having high cholesterol levels. It's essentially the additive effect, right? So my genotype at those risk variants. Okay, does that make sense? This is my risk, genetic risk. Okay, so let's say I care about cholesterol. Okay, so let's say a GWAS of cholesterol has given us 1,000 genetic variants associated with cholesterol levels. Okay, so my genetic risk will be my genotype at those 1,000 variants, weighted by the effect size of those variants. This is what we were calculating yesterday. Okay, this is how we calculate what's called the genetic risk score. You might, yeah.
By variants, do you mean tag SNPs?
Yes, by variants I mean tag SNPs, yeah.
So one variant can have multiple genes in it, so the number can be more than this? 100 variants with schizophrenia.
Yeah.
Then is it possible that there can be way more genes that are associated with it because you are tracking it with tag SNPs?
Are you saying, can there be more undetected variants? Is that what you mean? There can be more undetected variants, but now we've also gotten really good. I didn't speak about this at all. Even though we're only tagging, 1 million, based on this, we can statistically actually, what's in a process called imputation, figure out the genotypes of nearby variants because of linkage disequilibrium. So we're actually testing, we have information for more than 100 million. What we are missing is let's go back to, I really like this slide, but what we are entirely missing this, right? What we are missing is this entire half. So that's what we're missing. Does that answer your question? Okay. Okay, so that's, and some might say, okay, well, we always knew traits were polygenic. What else? Okay, but how are there so many risk factors, right? How do we connect this to biology, to what's actually going on in the cell? What we think is going on is that your phenotype has many, many genes acting upstream of it. A genetic change at any step of this pathway, which are often really complex, can lead to a change on the phenotype, right? So what we often see is, for a particular phenotype, we see changes, or we see the genetic variants associated with that phenotype have some sort of connection with the pathways upstream of that phenotype, right? Which is why they're all not concentrated on one gene. They sort of tend to be concentrated on all of the components of the pathway affecting that phenotype, yeah? Also, what we're seeing is that most of this causal variation is non-coding. Again, this was a bit of a surprise. We think of proteins as being functional, anything outside as not particularly functional, but that's not true, right? 90% of the results we see from GWAS are in the non-coding parts of the genome. Maybe you can also, maybe you can also sort of explain this with selection, right? You would not find common variation in a protein coding gene because there would be too much selection against it. Effect sizes of non-coding variants are more likely to be smaller, and therefore, most of this GWAS risk is concentrated in that regulatory part of the genome. Yeah.
The word causal, so
Huh.
I thought it just tells us association.
When I say, yeah.
Can we say causal?
So at an association signal, 90% of them do not have like a missense or a loss of function variant in that linkage region. So even if we don't know what is causal in that region or what is the actual cause, we know it's not a missense or a loss of function variant. Does that, but yeah, maybe I could have rephrased this by saying most associated signals are likely to be non-coding. Yeah, I think we're talking about the same thing, yeah. Okay, so what do we think again is going on? It's not that these causal variants are unlikely to be in these protein coding parts of the genome. They are way more likely to be sort of in a regulatory part of the genome, right, which don't control the actual protein being produced, but they change the levels of your mRNA molecules, the levels of protein being produced, right? So they are likely to affect gene regulation, gene expression, maybe protein translation. This is an area of active research right now. Yeah.
They do these studies, like especially in an Indian context, regarding caste system, like within caste marriages or cousin marriages in South India.
What about it? I mean,
These kind of samples.
So you're asking if these GWAS have been done in the context of consanguinity? So these studies tend to be focused on, as I said, common disease, but also unrelated populations, for a reason I will come to at the end. To study sort of, that's a really good question, right? That what happens when there's consanguinity? What happens if there's more genetic relatedness in the samples you're studying? We'll come to that at the end. But these are generally done in unrelated populations, and I'll tell you why, Okay. Another thing we see is that your GWAS signals tend to be enriched in tissues of interest. So if I pick a gene that's close to my GWAS signal, these are the GWAS signals for lipid levels, And on the y-axis is the enrichment of this GWAS signal in genes that are expressed in a given tissue. This is for, the labels have gone, sorry, but for LDL on the left, which is your bad cholesterol, we see the strongest enrichment of the GWAS signal in genes that are expressed in the liver, which makes sense, right? Cholesterol is metabolized in the liver. For HDL, which is the good cholesterol that you hear about, we see the strongest enrichment in adipose or fat tissue, which again is something we know about, right? So our GWAS signals, it's kind of cool. Your genetic variants are common across your genome, across all cells in your body. These genes are dynamic. They're differently expressed in different tissues of the body. But your GWAS signals for a particular trait are more likely to be found in those regions of the genome that are active in a tissue related to that trait. That was a very long sentence, so tell me if that wasn't clear. But let me illustrate that with one more example, This is one of my favorite examples. Let's look at this one, You take all of your GWAS signals for BMI, body mass index, and see, where do they lie? Where in the genome do they lie? Are they likely to lie near genes that are expressed everywhere? Are they likely to lie near genes that are expressed in the stomach, fat tissue? Where are they? What we see is that the strongest enrichment for your BMI signals is actually in the central nervous system, right? It's actually in the brain-related cell types. Yeah. These variants are no, these are mostly non-coding. There are ways to connect the variant to the gene, right? These variants are close to genes, or there are other functional assays we can do to try and predict what gene is this variant regulating.
No, because you said enrichment.
Yes?
How are you finding the enrichment if it's not in the gene? I mean, the number of mRNAs.
By enrichment, I just mean that they overlap, right? Do I have that slide? Yeah, okay. By enrichment, I mean this, right? Let's say these are your variants. The red bars are just the GWAS variants that you find. They are more likely than by chance to overlap with, let's say, active parts of the genome, right? So that's what I mean by enrichment. They're not randomly distributed through the genome. They're more likely to be found maybe in an active histone marks, and less likely to be found with repressive histone marks. I mean a statistical enrichment of overlap. Yeah. Is that clear? I think I lost a lot of people here, so let me go back to the previous slide. Okay, was this clear? Where our GWAS signals are mostly non-coding, they regulate genes, and we're able to find out the tissues in which these genes are regulated more likely than not by these GWAS signals, okay?
Arthritis and lupus, I understand.
Yeah,
Your immune system.
correct.
BMI, why?
Yeah, because one of the strongest signals for BMI is that hormone, is it leptin? The hormone that dictates satiety, whether you feel full or not. Yeah, so that's actually one of the strongest signals for BMI, which was a very cool finding. Yes, promoters, enhancers, all considered non-coding. Yeah. Absolutely, yeah. These are very important in regulating gene expression, right? Yeah. Okay. Okay, so what we also see is that, I'll go through this again. Let's say these are all of the signals that you find in a GWAS, right? There are other studies, these are called epigenetic studies, functional genomic studies, which try and measure which parts of the genome are active, okay? So let's say you take a liver cell, you do an experiment called a ChIP-seq, which essentially uses antibodies to pull down histones with a certain modification, and you have atlases, right? The advantage of, again, working with human genetics is you have these functional atlases of the genome. So this would say that, okay, this part of the genome seems to have some active histone marks around it. This part of the genome has some negative, repressive histone marks around it. And what we see is that these GWAS signals are very strongly, very, very strongly enriched in these activating marks, okay? Okay. One of the biggest challenges of GWAS is what's called population stratification. And I'd like you to sort of imagine what would happen if you ran a GWAS like this, okay? So let's say you care about, let's say, pick your disease, diabetes. Your cases, this is how you sample cases, just by accident. 75% of your cases are from northern India, 25% are from southern India, okay? And your controls is flipped. Would this work? What would your results look like? Let's say that's not really the case. The predisposition to diabetes is the same across the country, let's say. But just by chance, right, because of sampling. Let's say you just put more sampling stations in the north for cases and put more sampling stations in the south for controls by mistake. Or what would happen? Okay, hint, it's a bad thing. You shouldn't do this. But why should we not do this? Yeah, the ratio of these two populations is different in cases and controls, but so what? Why would, yeah, so you would get false positives, yes, but why would we get false positives? I mean, you're right, we would get a lot of false positives.
But there are more cases in north, but you are taking less samples from north.
No, we're taking the same number of samples from the north.
There is more prevalence in north.
Huh. We don't know, there's no method, the prevalence is the same throughout the country, let's say. Right? Yeah. Huh?
False negative.
It would actually give a lot of false positives, and I'll tell you why, right? It's because what you're doing is you're treating this whole group as cases, this whole group as controls, but between northern Indian populations and southern Indian populations, there's a whole bunch of other genetic differences, phenotypic differences that have nothing to do with diabetes. Your cases and your controls are not matched, either for your genetics or for your phenotype, right? Which means that you will get a lot of associations, differences between cases and controls that are actually differences between northern India and southern India. But you will think these are differences associated with diabetes, okay? And this is called population stratification. And it's a really, really difficult thing to solve in human genetics because it's not just northern versus southern, right? It's also slight differences in ancestry, which could lead to this false positives. And at some point, it becomes very difficult to tease apart if there is what's called population structure in your data, okay? Make sense? Okay. So if we actually, one way to visualize population structure is using what's called principal component analysis, okay? So if I take my genotype matrix, samples by SNPs, and I want to sort of just see how close are some samples in genetics, how far away are some other samples, we can somehow use the statistical technique called PCA, principal component analysis, to reduce the number of dimensions of your genetic genotype matrix into a much smaller number of dimensions, okay? So PCA is a dimension reduction and a visualization technique that captures, again, the distances, the genetic distances that are present in our samples. And this is what a PCA plot of human populations look like, okay? So again, PC1, which captures the most variation in your genetic data, separates Africans from all other populations. So this was what we discussed last time, right? The most, Africans are the most sort of distinct human populations. And along PC2, you see sort of Europeans separate out from East Asians on the right, the South Asians fall somewhere here, and you sort of see this gradient here, right? And even within India, if you plot only Indians, you would again, and made a PCA, you would see different regions of the country separate out. In India, there's also castes. You also see different like caste groups because of endogamy. You also see that separate out on a PCA, right? So you have to take population structure into account when we're running any kind of genetic study, yeah? Okay, so yeah, so that was PCA. And how do we bring that into our GWAS? Earlier, we had seen these linear regression models where we can include sex and age as we can correct for other factors. We can also include these principal components. We can include them in our analysis to say, okay, I want to correct for the effect of population structure or relatedness, right? So we bring those into our models. And in the tutorial today, I think, yeah, we have 5 minutes, but in the tutorial today, we'll sort of figure out how to use either R or a software to estimate these principal components and to try and run a toy GWAS, okay? Okay, how do we know we have corrected for population structure? It's by looking at these plots called a QQ plot, which is a complementary plot like the Manhattan plot, okay? So again, each dot, let's look at the one on the left. Each dot is a SNP that is plotted, okay? X-axis, what is the expected distribution of p-values you see under a uniform distribution? If you don't expect any genetic signal in your data, what do you expect your p-value distribution to look like? That's what the x-axis shows is, okay? It's a uniform distribution. The y-axis is the observed distribution of p-values, okay? If you have a lot of false positives, this is chi-squared, but we usually plot uniform distribution. We usually plot for uniform, yeah. The distribution of p-values for a random distribution is expected to be uniform. For no, right? If you just pick sample p-values from a random distribution, expected to be uniform. Yeah. Okay. Okay. So because of the enrichment of false positives, we see this line or the observed p-values above the diagonal, way more significant than by chance. On the right side, we have accurately corrected for population structure. This is a study that's been done well. Most SNPs don't show any more significance than you would expect. A few SNPs at the tail, these are actually significant, so you see them lift up from that background distribution, and we trust that these are actually significant because we also see that most of the genome is not significant, right? Here, it looks like most of the genome is behaving, giving you better results than you expect by chance, which means you're probably not doing some correction properly. There's some bias in your data. Yeah? Yeah. We will do it in the tutorial. These are two different GWAS analyses, one without correcting for population structure, one after having done a correction. Not the same one, no. These are two different ones, yeah? Okay. Okay. The gray is the confidence interval of the expected, yeah. Okay, so where are we now with GWAS? In the last 2 minutes, I'll just talk about the UK Biobank, which is earlier, in the first decade of GWAS, what we would do is, if you're interested in a disease, you form a consortium, you collect cases, collect controls, run your GWAS, publish a paper. Another person interested in a different disease, collect cases, collects controls, run a GWAS, publish a paper, blah, 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 right? Hundreds of papers published. But in 2018, the UK Biobank released this massive database of records from the National Health System in the UK This is the advantage of having a centralized health system where all medical providers in the country sort of put their data into this common database. And they have a few 100,000 individuals with genetic data, with phenotypic data for a bunch of diseases, all available to researchers to do anything on. So now we have most researchers are in some way working on, or most human genetics researchers are in some way working with this gigantic UK biobank. And the field is moving from sort of phenotype-specific cohorts to these biobanks, right? Working with hospital systems, working with national healthcare providers to study multiple phenotypes together instead of just creating these different cohorts for each phenotype. Okay. The challenges currently are, very active fields of research are, what do you do after you have a GWAS? What do we do with all this information? How do you make sure that your GWAS from one cohort is generalizable to another population? Let's say your GWAS on Europeans works on South Asians or vice versa. And the goal for a lot of people is to bring GWAS into the clinic, which is a very far away goal. But for cardiovascular disease, and I'll end here, it's actually beginning to be implemented, okay? So I'm just gonna point you to the figure on the right where you run GWAS on cardiovascular disease, right? It's coronary artery disease or CAD. And what we do is you estimate all your variants, calculate genetic risk for a new bunch of people, and sort those people by percentile of genetic risk, okay? So people who are least likely, according to their genetics, to have cardiovascular disease, people who are most likely, according to their genetics, to have cardiovascular disease. This is independent of their phenotype. We have no idea. We are blinded to the phenotype at this point. And on the y-axis, you plot, okay, in this group of people, what is the prevalence of what is the actual prevalence of disease? If you now go and look at their phenotype, what is their prevalence? And you see, right, these risk scores work to some degree. I mean, the people in the top 10 percentile of genetic risk are, I think, 4-fold or 5-fold more likely to have a heart attack than people in the bottom 10 percentile, right? So genetic risk is indicative or informative for actual risk. For something like cardiovascular risk, for lipids, knowing individuals in this 10% could actually benefit from knowing that because there are early interventions. You can use statins, you can bring in lifestyle interventions, you can bring in diet interventions early on before diagnosis, right? So this is this is the only phenotype for which genetic risk is even close to coming into the clinic. For any other phenotype, we're nowhere close, but this is sort of gives us it's a bit of a promise that it is possible, yeah? Okay,
Yeah, so I think population stratification, you said we are likely to overestimate effects.
Yeah.
Like for human height, I think it happened, right?
Correct.
So why is it always overestimating? You said more false positives. Can you explain why?
More false positives because we are more likely to pick up differences between cases and controls. There are actually differences because of genetic relatedness or...
Why not more false negatives also?
Why
Is it in this one direction? So it's not clear to me.
Well, false negatives would not be inflated by the cases and controls being genetically more unrelated. Your false positives, because you're still comparing cases and controls.
Yes.
So your false negatives would not be affected by this bias. Maybe we can write it down and do it on paper. I think that'll, yeah. Okay. All right, so we'll go for lunch and see you in an hour.